Thank you for giving me the opportunity to present my work. The title of my talk is Investigating Stability and Dynamics of Proteins Using Molecular Dynamics Simulations and Effective Analysis Method. I'm interested in the mechanisms of protein systems from the viewpoint of stability and dynamics using all-atom molecular simulations based on physics and chemistry. Especially, I have developed algorithms related to protein simulations. There are three main challenges in conducting protein molecular simulations. First, challenging is sampling. Maybe about 20 years ago, I developed algorithms for generalized ensemble algorithms. I formalize enhanced sampling method and demonstrate the effectiveness using simple system. Today, related methods are widely used in protein simulations. In particular, I developed method related to simulated tempering. This method reduced the number of replicas required, making it well suitable for GPU-based computing. I expected it will become even more widely adopted in future. Enhanced sampling methods are applicable not only to protein system, but also many other systems, like molecular systems. Recently, I have developed analysis method, and for investigating stability of proteins, rhythm theory, maybe previously presented. And also, I developed dynamical analysis method, relaxation mode analysis. We are now working on understanding protein functions at the atomic level using this method. I also conduct applied research in close collaboration with experimentalists. I little bit explain to investigate the stability of proteins. Usually, there are competition between conformational energy and solvation energy. Proteins achieve their stabilities from the balance between conformational energy and solvation-free energy. It is important to estimate the solvation effect. Rhythm theory is a method based on statistical mechanics of liquid. And if we solve the equation self-consistently, we can obtain the distribution of solvent. We collaborate with Dr. Maruyama, who is the expert of rhythm theory. Dr. Maruyama has worked on accelerating rhythm program using supercomputer of Fugak and Tsukuba Supercomputer Center. And then now we can calculate quickly and stably using his program. This is not my work, but Dr. Yoshida and also Dr. Maruyama now developed an open source package called Rhythmica. It's an excellent program. By using the programs, we investigated that kind of programs. First, we investigated the stability of folding trajectories calculated by Anton, including solvent effect. The total energy consists of solvent effect and structural energy. By analyzing folding data, we found the balance between conformational energy and solvation-free energy are important to achieve native structure. This is native structure. Native structure have lowest total conformation, total energy. And also, we investigate the energy stability at amino acid level for stable and misfolded structure of synuclein 10 residue peptide using atomic decomposition method proposed by Hamson. From this analysis, we can investigate the stabilities, amino acid levels. From this result, we propose amino acid substitution to stabilize misfolded state and solve the structure by NMR. And then also, we apply this method to analyze RBD, ACE2, and also folding trajectories. And then we a bit move to the analysis method. Background of analysis method is here. We want to extract important modes from complicated motion of proteins automatically. We want to reduce multiple degrees of freedom of complex motion of protein to lower dimensions and extract characteristic structure change. We want that kind of axis. If we obtain that kind of axis, we can calculate free energy surfaces and then investigate the conventional changes. Recently, very long simulations have been feasible. Then we need dynamical analysis method. We develop relaxation mode analysis. Relaxation mode analysis have been developed for finding slow motion in random spin system and homopolymer system. We apply this method to heteropolymer system. Relaxation mode is here. Relaxation mode satisfies the equation. This is relaxation mode, and this is relaxation rate. Relaxation time is inversely proportional to relaxation rate. From the trajectory simulation data, we extract modes with slow relaxation by using relaxation mode analysis. We approximate relaxation modes like that, and then considering variational problem of here, we finally obtained generalized eigenvalue problem of time correlation functions of coordinates with two different times. By solving the equations, we can obtain modes with slow relaxation. In 1995, Professor Takano, who was my colleague at Keio University and now is Professor Emiti at Keio University, developed this method in spin systems. And then also, he applied this method to polymer system. Polymer system, he investigated behavior of slowest relaxation rates, which is good agreement with the theoretical prediction. We apply this method to protein systems. And then also, we develop methods related to relaxation mode analysis. Here, I little bit introduce the demonstrate the effectiveness of this method by applying this method to following trajectory data. Before showing the result, I little bit explain the difference between PCA and RMA. PCA is maybe famous principal component analysis. This method extracts modes with large variance. Relaxation mode analysis extract modes with slow relaxation. If there is a free energy surface, PCA extracts this axis with large variance. In that case, the 2 points can't distinguish. But RMA, if there is a higher energy barrier, the motion corresponds to the slow motion. Then RMA extracts this axis. In that case, this method can distinguish this state and this state. RMA is suitable to analyze protein folding and intrinsically disordered protein system. And then also RMA identifies slow conformational changing with slow transitions. We obtained the trajectory of folding simulation data that previously published by asking the show group this data. The data, maybe they show, said this data, maybe two holding passes are in there, but they don't explain in details. We analyze this data by using relaxation mode analysis. And this is slowest relaxation mode axis. This is second slowest relaxation mode axis. In that case, RMA can distinguish that kind of states. If we use PCA to the data, PCA cannot distinguish that kind of state. RMA, we little bit colored characteristic states. If we put these states on PC surface, maybe here part, there are many overlaps, we cannot distinguish. And also, these are spread widely. While it was difficult to identify characteristic state by PCA, RMA clearly classified it, not only structure with small difference, but also structure which is partially folded. And then if we analyze very carefully, we can find two folding pathways clearly. If we obtain the good order parameters, we can analyze the folding process. Here, maybe here case, this pathway, maybe here case from here from to here. And then recently, we want to use this method to conduct applied research in collaborating with experimental researchers. Our target is GPCR, G protein coupled receptor. 30% drug discovery targets for small molecule ligands are GPCR. And then my target is orexin-2 receptor. Orexin-2 receptor is one of GPCRs. This is involving in regulation of sleep-wake cycle, and development of agonist for orexin-2R is expected as a therapeutic treatment for narcolepsy like that. Yeah, GPCRs are membrane protein like that. 7 transmembrane helix. And then GPCR have active and inactive state. This is inactive state. This is active state. If the agonist binds to this site, GPCR change conformation to like that. And then this protein have wider space near there. G protein binds to here. And then function may be cascaded. Signaling is cascaded. And then also there are two pathways, a G protein pathway and arrestin pathways. In that case, we need to molecular dynamics simulations because these are a small difference. Maybe we need to investigate the atomic level information. And then structure, there are several tough work, structure determination. Membrane protein and structure determination are much difficult. But recently, structures of inactive state have been reviewed by X-ray crystallography. And then also recently, structures of complex with G protein in active state has been analyzed by cryo-EM. This one and also this one. Yeah, these structures are solved by experiments, X-ray and cryo-EM, but before binding G protein, the state of active state is unknown. MD simulation can investigate the active state by removing G protein. Also, MD simulation can capture activating process between inactive and active states. We have two works for orexin 2 receptor. Several microsecond MD simulations from inactive states using constitutively active mutants. This mutant may be no ligand. This structure changed to active structure, which show are active without ligand, and introduced in other GPCRs. And then we maybe catch the activation process by using the mutants. And then also we perform several microsecond simulations of inactive and active structure without ligand. Sorry, no. We prepare 5 systems in active state with ligand and remove ligand. And then also this structure data, previous work, we obtain the structure change from inactive to active. And then also active ligand binding active structure and ligand-free active structure. Maybe these kind of huge MD simulations are performed. And then analyze the trajectories by using relaxation mode analysis. This is the slowest relaxation mode axis. From here, we can find this axis maybe correspond to structural difference between inactive and active state. These are inactive states data. These are active states data. This axis distinguish between them. RMA also can get the information of movement along the relaxation mode. Then we calculate the collective motion of TIM domains using during activation. We can capture the collective motion of TIM domains, and from this motion, we investigate the connectivity from the ligand binding site and to protein binding site. Also, the important residue to activation can be also investigated. This is TIM3 have a typical motion, upper motion. In that case, the 3 residues have high structural changes. Then we analyze the residues deeply. And from here, we little bit found by identifying amino acid that exhibit characteristic movement and analyze their dynamics in detail, we can propose interaction important for activation. These are a summary. Sorry, I skipped. And then recently, we started to perform simulation with various ligands using structure obtained by X-ray, cryo-EM, we want to investigate function of proteins by analyzing dynamics of protein using molecular simulations. These are the collaborators for experimentalists. Also, we started to simulations, molecular dynamics simulations of like that kind of systems with collaboration for experimentalists. With one, I don't say that. There was liposome. Liposome case, we investigate dissociation of IF1 from the coordinating center here. From the we obtain the structure from the experimentalists, and then we perform simulations that kind of huge systems. And then we investigate dynamics these systems. Sorry. Summary is here. We developed and introduced algorithms related to Meiji simulation, and then also we use this method to investigate dynamics of protein simulation. And then also we recently used we collaborate with experimentalists to investigate functions using molecular dynamics simulations. Advances in experimental techniques, computers, and methods of MD simulation have brought the timescale of experiments and simulation closer together. Especially supercomputers such as FUGAC support the large-scale simulations of biomolecular systems. It is believed that new discoveries will be made through joint research. These are the collaborators, and also these are collaborators for experimentalists. So thank you for your attention.